Hi, and thank you for calling into today's webinar. This is part of the SSPC webinar series and is being first broadcast on September the 18th, 2020. The webinar is on the continuous crystallization of active pharmaceutical ingredients, and I will be delivering today's webinar. My name is Stephen Ferguson. I'm currently an assistant professor at the School of Chemical and Bioprocess Engineering in University College Dublin, and I'm also the team lead for manufacturing as part of the SSPC Research Centre. This is the Science Foundation Ireland Research Centre for Pharmaceuticals. Over the course of today's webinar, we'll of course discuss continuous crystallization of active pharmaceutical ingredients. And to this end, we'll first discuss the types of continuous crystallizers that have been used in the scientific literature and in the industry to date in isolating APIs continuously. Furthermore, we'll discuss the dynamics that these different continuous crystallizers offer in comparison to batch isolation or crystallization, which is the current industry standard, and how these additional process trajectories or crystallization dynamics offer the ability to control and also to access a wider range of critical quality attributes for active pharmaceutical ingredients, namely here the mean size, the morphology of the particulates, the polymorphic or crystalline phase that's isolated and its purity, the chemical purity of the API, and finally the chiral purity or the enantiomer of the API that can be isolated as well. So before commencing the presentation, maybe just a brief note that all of the case studies used and all of the literature that is discussed at a high level relates to developments over about the past decade or so, wherein continuous crystallization, which was originally a topic of research in the 60s, but effectively didn't see significant research for a number of decades, essentially experienced a spurt of activity based on the need to develop continuous crystallizations for active pharmaceutical ingredients. And this was driven by a wider development effort to develop flow chemical and continuous pharmaceutical processes to intensify the production of continuous pharmaceuticals and fine chemicals. These molecules are somewhat different to the commodity chemicals that were investigated in the 60s, things like potash crystallizations, which were very small molecular weight compounds isolated on very large scales in manufacturing, compared to the current research efforts that have much more highly functionalized organic molecules isolated on much smaller scales, which presents very different challenges and resulted in some different approaches being taken and developed over the past 10 years, and that will be the focus of what will be outlined. I'll put up some references to some textbooks that do outline this earlier development, and there's some seminal works that will be put down in the reference section from this earlier large-scale efforts to produce rational design for continuous crystallizations for large-scale commodity chemicals. To begin, we'll look at the types of continuous crystallizers that have been commonly used in the crystallization of active pharmaceutical ingredients. So of the types of continuous crystallizers, the most commonly used for active pharmaceutical ingredients are stirred tank type crystallizers, and in particular MSNPR type crystallizers. These two terminologies are often used interchangeably, but there are some constraints on the way MSNPR type crystallizers operate. A common example of this is shown from the paper conducted here below in the SSPC in UCD. This is a Optimax stir tank reactor that is temperature controllable. To measure concentration in situ, there is FTIR, and to measure the particulate characteristics of the ongoing crystallization, we placed in an FPRM probe as well here. Apart from that, there's nothing really too complicated involved in configuring a standard lab batch reactor or vessel to be a continuous crystallizer for the purposes of optimization or lab scale trials. So two small peristaltic pumps were used, one to pump in feed and one to pump in anti-solvent. And when these are brought together in this mixed vessel, this generates supersaturation for crystallization. If we just pumped in feed and anti-solvent, the vessel would overflow, it would flood. So there is the necessity to have an outlet here. In this case, a pneumatic transfer system was used, and this was used for a couple of reasons. So number one, the MSMPR type crystallizer means that we assume that we have mixed product removal or idealized removal. So this means that the particulates, the crystals that are stirred in here, are removed in a representative way, meaning that, for example, the small particles are preferentially sucked out as they're removed, leaving the larger particles to have a longer residence time in here. So the particles of all sizes have the same residence time, they have the same chance of being removed. And additionally, there is another assumption for MSMPRs that they're perfectly mixed, so meaning that the liquid phase concentration is homogeneous throughout the vessel geometry. And also that if we were to measure the particle size, we were to put the FBRM probe in at any different point, we would get the same measurement of particle size and number of particles, solids density, whatever particulate properties or solid phase properties we were to measure of the slurry at any point would be the same also. So those are the two constraining assumptions of MSMPR type crystallizers. There are other types of continuous crystallizers that, for example, allow the fines to be dissolved or allow only particles of a certain size to escape from the crystallizer. But MSMPR type crystallizers essentially ensure good mixing, representative withdrawal. And this can be achieved or approximated, these assumptions on a variety of scales. So to ensure correct withdrawal or representative withdrawal, so that we don't have large particulates that will stay in, grow, and agglomerate, eventually causing problems for the system. A technique of intermittent withdrawal is used. So this allows, in the case of this study, was the demonstration for a pneumatic version of this, having a fill of about 10% or up to 10% of the vessel volume, and then every 1/10 of a residence time, turning on a pneumatic transfer mechanism. So either this can involve pressurizing the vessel or pulling a vacuum, in this case on a filter, which is downstream, as this is only a single stage. So typically the residence times in MSMPRs will be of the order of minutes up to hours, and we would do the withdrawal over approximately 2 to 3 seconds, whereby a pressure difference would be created. Fluid would flow up through a dip tube and out into a filter where it was collected and the samples were analyzed. Fluid or the slurry would flow out through this dip tube until the minimum level of the dip tube was reached. In order to keep the system clear and to prevent blocking of this outlet line, which contains supersaturated solution, as is a feature of MSMPRs, the transfer mechanism was left on a little bit longer, which allowed air in this case, but normally nitrogen, to pass through the system and flush out this line. So apart from the 2 to 3 seconds where transfers are taking place, and it's often done much faster than this, the line will be empty. So there wouldn't be the possibility of supersaturated solution crystallizing in this line and blocking it. And so this was a key to allowing prolonged run times at small scales of operation. We've mentioned already that a lot of work was done on continuous crystallization in the 60s and 70s, and then there was a gap until the recent development of the technology for pharmaceuticals in particular. Well, if you did look at that previous literature, you were to go to, say, Mullen or Yaroslav Neivelt's textbook, which summarizes older crystallization technology development. They wouldn't recommend running crystallizers at this scale, particularly for manufacturing purposes. And one of the chief concerns will be the reliability. So a lot of the practical aspects and know-how have been key in progressing this field forward. And there's a couple of papers that I referenced that outline techniques that allow this to be done. A very commonly used strategy is to not use one MSMPR type crystallizer, but to group together multiple MSMPR type crystallizers in what's known as an MSMPR cascade, which is essentially connecting multiple MSMPRs together. So highlighted here is a schematic of a pneumatically driven transfer system for a multiple MSMPR system cascade, in this case a 2 MSMPR cascade. Again, this is conducted by Brian Glennon's research group in SSPC in UCD. In this case, the configuration has a feed and product tank that go to the same location. And this is another common strategy used at lab scale to limit the amount of material needed for development. So rather than having to use a lot of material to cycle through the system until it will go to steady state, you can dissolve the product as it comes out and allow a steady state for a given set of conditions to be achieved in the MSNPR system. And then you can alter some of your process parameters and see what impact this will go on to have on the steady states and the particular properties of the crystal size distribution that are isolated. So this is quite a common development approach used to optimize continuous stirred tank type crystallizers in particular. Another strategy that is used practically when running these is that the transfer lines between vessels and also the feeds going into the vessels often will use heat tracing of the lines or possibly double jacketing also. So you can take the lines that go in from, say, a feed tank here and place a larger pipe or tube around, and you can use a pumped heater chiller that are very commonly available in lab settings to pump hot fluid around. You can link all these together and you can temperature control the transfer lines and the feed lines at above the supersaturation, above the temperature where supersaturation occurs for the given system that is being tested. So that is another strategy. Again, this uses pneumatic transfer in order to keep the lines that have supersaturated slurries clear apart from the small period of seconds in each transfer when the material is being pushed through the line and then cleared using gas. The ability to line the pipes that connect the vessels, it's often not used that frequently when you're doing transfer between vessels, particularly when you purge the lines, as they don't typically have for any prolonged period of time any supersaturated solution in there or present in the line to allow crystallization occur as material are transferred across. So I should also give note to Eli Lilly, who did a lot of work pioneering the use of pneumatic transfer in both heterogeneous reactions in continuous flow between stirred tanks and also for crystallization purposes as well. This describes the development and the first publications in literature that were conducted by the SSPC over the course of a number of PhDs in Brian Glennon's research group, including my own. So we'll discuss in more detail when we're going through reactor dynamics on some of the case studies, the impact using multiple stirred tanks will have on the product qualities. However, from a practical point of view, there's another important thing to note. So if I have a feed solution being brought into an MSNPR type crystallizer, for example, if I'm using cooling to generate the supersaturation, so I have a hot feed tank, whereby material can stay clear with everything dissolved in solution at maybe 80 degrees. And I want to have a final isolated yield of, say, 90%. I may have to cool this down to 5 degrees. So this can be practically something difficult to do because of heat transfer limitations in stirred tanks. But also, even in cases where we do have sufficient cooling capacity on the jacket, this may result in a very low jacket temperature compared to the bulk temperature within the crystallizer, which will have to take out the heat that's put in when the material is pumped into the vessel, and also the heat of mixing and crystallization. And so from a practical point of view, this will tend to lead to growth and fouling of crystals upon the walls relatively rapidly. But by using multiple stirred tanks, we can spread out this burden in terms of cooling. And so this will allow, say, a smaller temperature drop. So if we wanted to go from 80 degrees to 5 degrees, you can now take a 40-degree drop and then another 40-degree or another 35-degree drop, as opposed to just taking it all in one go. Similar constraints may exist with respect to mixing, putting in an entire anti-solvent charge in one stirred tank, although that will depend on the residence time and the degree of the charge. But this is primarily a question in terms of cooling crystallization, in terms of practicalities around fouling and blockage. By splitting the supersaturation amongst more vessels, on average you can operate at lower supersaturations and generate larger particle sizes, and also in the case of cascades you'll get narrower particle size distributions than when only a single stir tank crystallizer is used. Overall, those stir tanks are very popular as outlined. They allow high solids loading. They are quite robust compared to alternatives, and they have a lot of flexibility in terms of the residence times and the product qualities that are required in API crystallization. Following continuous stirred tank type crystallizers and cascades of continuous stirred tank type crystallizers, tubular or plug flow type crystallizers would be the most commonly utilized platform for the crystallization of APIs. That said, despite being quite well represented in literature, they're less commonly taken up as within the pharmaceutical industry, at least to date, for the purposes of crystallization, at least. So there are a number of different varieties of continuous tubular or plug flow type crystallizers, the simplest of which is just a pipe or a tubular section. So either anti-solvent and another feed stream can be brought together and given residence time within a tubular crystallizer. If enough residence time is provided, so at a given flow rate, if a long enough tube is provided, the system can go to thermodynamic equilibrium. Similarly, the tubular section can be cooled in the case of cooling type crystallizations. So as you can imagine, if your crystallizer is simply a pipe, this is quite cost effective, modular, simple. So why are these not the most common platform? Well, there are a couple of driving factors in this. First of all will be the relatively long residence times required for the crystallization of active pharmaceutical ingredients. As organic, relatively highly functionalized, large molecular weight molecules, they are more prone to have more polymorphs than the commodity chemicals that would have been investigated in the development of continuous crystallization research up until the focus moved to pharmaceutical ingredients in the past decade or so. And so very aggressive cooling ramps can result in the isolation of amorphous materials or high energy polymorphs that are typically not desired for use as active pharmaceutical ingredients that, where possible, will seek typically to look to isolate the most thermodynamically stable form. In addition, the throughput requirements are relatively low. So to produce the amount needed for the supply of many drugs for the entire world will not typically, in large tubes, if run on a year-long basis, have high enough flow rates to generate turbulence. So turbulence occurs when at higher flow rates above 4,000 Reynolds number, we see the onset of turbulent flow. And in turbulent flows, the crystallizer will effectively mix itself. It will have limited mixing up and down the length of the tube, but it will have very good mixing in the radial direction towards the edges of the tube, which gives good mixing and heat transfer characteristics that are spontaneously generated in very simple tubular equipment. So typically these setups can be used when low residence times are required. In the case of crystallization, we do also have to suspend the solid particles. So turbulence at higher Reynolds numbers can do that spontaneously. This is not the case for very slow laminar flows where the particulates will tend to sediment and are usually avoided for the cases of crystallization. In order to approximate the attributes of plug flow in tubular crystallizers with reasonable diameters to avoid blockage and with lower throughputs that would ordinarily have laminar type flow with poor circulation and ability to suspend particles, a number of alternate approaches have been proposed for tubular crystallizers, and two popular ones are shown here, and this is using a graph that was reproduced by a review of continuous crystallization by Zhang et al. And in this graph, we see a segmented flow crystallizer and an oscillatory baffled crystallizer. So both of these essentially aim to have the benefits of plug flow while operating at lower throughputs that would ordinarily have laminar flows, but can do two things. Firstly, operate at on reasonable scale crystallizers with throughputs that are more commonly used at longer residence times that are also typically used in pharmaceutical crystallizations. Very briefly, segmented flow crystallizers will introduce an immiscible fluid into the tubular flow, and hence the mother liquor or slurry as it begins to crystallize will progress in different segments, as the name suggests, with a segment crystallizing followed by an immiscible segment, another segment with crystallization volume. This approximates plug flow, as between the segments there is an immiscible fluid, so we can get limited backmixing or no backmixing from this segment into the next one behind it or in front of it, hence having something that looks more like plug flow type behavior. In addition to this, the laminar flow rates at which these segments are used, the flow or the laminar flow is interrupted by the presence of essentially this moving boundary, which does generate some circulation, improving heat transfer and allowing for some suspension of particles up to limited solids loadings. Hence, attaining some of the attributes of plug flow type crystallization at lower flows under what would normally be laminar conditions. Similarly, oscillatory flow crystallizers have a net flow along the axial direction from inlet to outlet that is low and will be characterized by laminar conditions. However, superimposed on this low net flow rate is an oscillatory flow that is much higher. So as this slow net flow passes through baffles, this generates mixing, hence suspending the particles and also improving the mass and heat transfer characteristics within the tubular device. In addition, there's limited mixing, although not no mixing, in the axial direction up and down. And as a result, with a large enough number of segments, of which there are usually quite a few within a continuous oscillatory baffled crystallizer, the crystallization begins to look like an idealized type plug flow that will be characterized by high Reynolds number tubular flow. So before moving on, just briefly, I've just put up a schematic of a plug flow type crystallizer that would be suitable for an anti-solvent here, but also a reactive type crystallization where two streams are brought together in a high intensity Rowton type vortex mixer. This vortex type mixer, which is in perspex here and in stainless steel here, then feeds into a tubular section, in this case, which was run up to a Reynolds number of 10,000 with some caveats, allows the crystallization to progress to equilibrium, and this can be measured in situ with some purpose-built flow cells and FTIR. We could also measure the particular properties in situ with FBRAM, which is showcased in this another study from the SSPC. While such a platform isn't likely to be a catch-all for a significant or complete array of pharmaceutical crystallizations, where they are applicable and you can increase supersaturation and run your crystallizations quickly, there are certainly some situations where there's significant utility. First of all, where we want to drive down particle size and make a small, narrow crystal size distribution. This may be useful for enhancement of bioavailability with a given crystalline form without milling, or in some cases for control of particle size distribution for pulmonary applications. In addition to this, one of the downsides may also be a benefit. So you may want to attempt to make amorphous forms or high energy metastable crystalline forms of your API, and high intensity, high supersaturation plug flow crystallizers may be a means to do this. They're also hugely productive when they are possible to run, as you can imagine, with very low residence times and very high throughputs for a very compact device. So supplementary technologies are often added to continuous crystallizers. For example, recycle loops, which we'll discuss further in terms of improvement of yield and purity in continuous crystallization systems. Wet mills, which are shown schematically here for continuous single-stage MSMPR crystallization, whereby the contents of a MSMPR are drawn out through a rotor-stator type wet mill, in this case a Magic Lab in this study by Kulgas and Vetter. That was evaluating the impact of wet milling on the ability of MSMPRs to control polymorphic outcome of a crystallization. This can be recirculated at a relatively high flow rate, and wet mills are very scalable in that the tip speed can be maintained from lab scales up to very, very large scale production. In addition to this, there is the possibility of continuous seeding, where you may recycle back from the end to the beginning of a plug flow type crystallizer. Obviously, with backmix MSMPR type crystallizers, this is less beneficial because they are completely backmixed within a given stage. And also there's been some fouling prevention strategies where automated in situ intervention is carried out in order to mitigate any fouling issues that may occur over the course of a crystallization. So before moving on to illustrate how continuous crystallization can be used to control all the critical quality attributes needed for active pharmaceutical ingredients, both from a processing and from a product quality point of view, we'll briefly discuss how these different types of crystallizers that we've outlined will allow for a different range of crystallization dynamics or process trajectories to be taken between the starting point, the feed of a crystallization, and the end point where isolation occurs. And in so doing, a wider range of particle size and product quality attributes to be achieved in comparison to when only batch crystallization is considered. In all these cases, the methodologies used to control to a given specification are outlined also. So the figure shown here is the solubility curve for an anti-solvent crystallization. On the graph, we have increasing anti-solvent composition in the system per unit solvent or on a per unit mass solvent basis. And on the x-axis, we have dissolved solute, again on a per unit mass or per gram solvent basis. So as we would expect, as there's more anti-solvent added into the solution, the solubility of the dissolved solute goes down, and as always, the solubility curve shows the thermodynamically stable amount of dissolved solute in solution. Superimposed on this is the typical supersaturation trajectory that is seen in the common types of crystallizers that we've discussed to date. And in all these cases, we have a fixed starting point, so a fixed composition of solvent, anti-solvent, and dissolved solute concentration. We also have a fixed end point. This is on the phase diagram, so this is at thermodynamic equilibrium. And so in all these cases, we're going to go from one fixed point or a feed to a fixed end point. So it's immediately obvious when we consider continuous crystallizers, MSMPR and plug flow crystallizers, we have a much wider range of paths down this phase diagram between these 2 points, a wider range of supersaturation histories that the solution and the slurry can experience, and hence, as we would expect, a wider range of sizes and product quality attributes. So let's start off in a typical fed batch crystallization where we have a stir tank and we add in anti-solvent. We start off undersaturated, so at this point, if we throw in any solids, they will dissolve. As we continue to add in anti-solvent, we move to the right on the diagram. There's no crystallization occurring until we reach the solubility curve, so we move horizontally. We then cross over and generate a supersaturated solution. It doesn't immediately nucleate until we get a burst of primary nucleation followed by secondary nucleation and growth, or until we seed our batch crystallization. At this point, there is a burst of secondary nucleation growth while at high supersaturation, followed by additional charging of anti-solvent, which maintains a roughly constant supersaturation in this case, until we reach the end of our charge when the addition of antisolvent is stopped and the concentration will drop to that at the phase diagram eventually, and the system will go to thermodynamic equilibrium. In comparison to the batch crystallization, in the case of a plug flow crystallization where we rapidly mix, if we mix faster than the nucleation can occur, faster than the induction time, we can jump immediately to this furthest point out on the crystallization. And hence all of the nucleation and growth and all of the time experience or residence time in the plug flow crystallizer occurs at this one fixed solvent-anti-solvent ratio. And we can see here that we are able to generate a higher net amount of supersaturation and access a higher amount of supersaturation, which is the difference between the actual solubility and the thermodynamically possible solubility if left for a very long time. And so we're able to maximize this, and so this will be indicative of a high energy plug flow type crystallization. We see that both the plug flow crystallizer, which will jump to a given point of the phase diagram and then will progress towards equilibrium along the length of the tubular section, and the batch crystallization, where antisolvent is added versus time, and after nucleation occurs or seeding occurs, we'll deplete supersaturation as we add as a function of time or antisolvent until we reach our end point for our crystallization. So the plug flow crystallizer will progress with respect to spatial position or length along the crystallizer. And the batch crystallizer will progress with respect to time. From the MSM pure crystallizer, this will operate at a single point on the phase diagram. So the MSM pure crystallizer operates at a steady state that is the same at all points in the crystallizer as it's well mixed, and as it removes the product from that fixed point, it only ever operates at one point on the phase diagram. So this is very different. So it's at steady state with respect to both time and spatial position. And this is true for all of the individual MSMPR crystallizers. And again, by using a cascade, we don't have to make the entire antisolvent charge in one go. We generate less supersaturation in a single vessel, and this will bias a system towards having larger average particle size, as nucleation is more favored by higher supersaturations than growth. In terms of kinetics, nucleation tends to have a higher order with respect to supersaturation than crystal growth. Just a couple of things to note. Firstly, that in a batch crystallization where the system has relatively slow nucleation kinetics, you may be able to charge the entirety of your antisolvent charge prior to nucleation occurring. And at this point, you can hold your crystallization, agitating it. Eventually nucleation will occur, and the entire nucleation and growth will occur as a function of time until eventually all of the supersaturation or additional concentration above the concentration of the solubility curve at this antisolvent-solvent ratio will be depleted as a function of time. Hence, the dynamics in this case will be exactly the same for both the batch and the plug flow crystallization case. For systems with faster nucleation kinetics, it will actually never be possible to charge fast enough to actually jump out to this point, as the system will induct prior to mixing. So the induction time for nucleation will be lower than the time for blending the two solutions together, even if you pour all of the antisolvent in one go to the agitated batch crystallization. For this reason, from a point of view of the crystallization dynamics or in terms of modeling, a batch where everything is added prior to nucleation occurring, and a plug flow where everything is mixed together prior to nucleation occurring. They're equivalent in terms of the dynamics, except one progresses with respect to time and one progresses with respect to spatial position. And as a result, the equations for the population balance for these are the same, or they're equivalent, again, except for the time in a batch crystallization replaced with residence time or length or volume for the plug flow type crystallizer. Similarly, if I take an MSM would operate at a steady state with respect to both time and spatial position, and I add more units to a cascade, I am bringing these more and more down to lower supersaturations and an increasing number of charges. And in the limit, if I add an infinite number of MSM I will have essentially a continuous or a continuum of anti-solvent addition. However, a larger number of MSM crystallizers will mimic the behavior of a batch or a plug flow type crystallizer where there is a fed batch or a time-spaced charge of anti-solvent. The crystallizers such as the continuous oscillatory baffle crystallizer will operate in a similar way to this, with each small baffled section operating similarly to an MSMPR within an MSMPR cascade. So over the next few slides, we'll look at how this impacts particle size distribution using a case study of benzoic acid. That was a test system that we use commonly in UCD as a benchmark to compare different types of crystallizers. And then we'll look at how these different dynamics can be used to control polymorphism of APIs in continuous crystallization, enhance the purification and yield in crystallization systems, and also to do chiral resolution continuously. So to begin with the case study of benzoic acid for the evaluation of a couple of different types of continuous crystallizers, we'll start by looking at how those crystallizers reach the steady states and what those dynamics actually look like within continuous crystallizers for a real example. And to this end, we'll start with the steady state operation or the achievement of steady state operation with a tubular or plug flow. And what does this really mean in practice? So I've highlighted the route and type vortex mixer section of the plug flow here. One thing to note is this is a very small device. I compared it to a small coin earlier in the presentation. In this device, you'll pump in feed from one side and anti-solvent from another side to generate high supersaturation and try and have a rapid crystallization. In this case, we're trying to engineer small particle sizes on the range of 1 to 20 microns. The volume here is quite low and the flow rates are quite high, so on the order of up to a liter a minute coming from both sides. So the residence time is approximately 16 milliseconds shown in this picture here, and we can see quite clearly that crystallization or induction, nucleation and growth begins before this nucleation, this 16 millisecond residence time is reached and the solution or the slurry flows out into the tubular section of the plug flow crystallizer. So in this case, in fact, there were actually physical or mixing limitations, and this is not really the core focus of this study, but we'll be happy to take any questions on this point. What we really want to focus on here is, what happens when we start up a tubular plug flow crystallizer? What should we expect to see? And when we say it has dynamics and steady state, what do those mean within a tubular type crystallizer? So we can look at this directly by putting a PAT device in situ. And so this was done by developing in situ PAT flow cells, which will sit at a fixed residence time. So if I take my vortex mixer and I attach it to a tubular section, I will have a residence time that will be set by the flow rate of the overall fluid coming in, and also the amount of volume or the length of the tube that is provided. So the plot here shows what is seen by an FBRM within a flow cell at a fixed point along a tubular section, and a FTIR, which measures the concentration of benzoic acid as it changes. And this is what happens from a startup when initially the crystallizer is filled with solvent. So it's pre-charged with a cleaning solvent that can actually, in the case of this crystallizer, be used to clean it out entirely in a few seconds. And we will bring in our feed and anti-solvent together to conduct the crystallization. So initially what we see is a purging of the cleaning solution, which is again an alcohol-based cleaning solution similar to the solvent in this case, which was ethanol. And we initially just see noise and the cleaning solution. This is very quickly washed out, and the crystallizer begins to go to a steady state. The steady state is achieved very quickly. It's actually just the scan time of the instruments was the limitation in how things go to steady state. In theory, if a plug flow does have true plug flow type flow patterns, on the order of 1 or 2 residence time should be enough just to displace the material from the crystallizer and reach steady state. This would seemingly take longer here as we purge out our material, but again, it's the scan time of the instruments, which are typically set to 15 to 30 seconds for the FPRM and the FTIR in this case, although they are set at their lowest settings here, which are 15 seconds and 2 seconds in the case of the FPRM. So we see that we very quickly achieve a steady state, and we see that basically it becomes quite boring from here. So if we leave this in theory pumping forever and generating crystalline slurry, we just operate at a single size in terms of the particulate mean size. At a given point there is nothing happening. So when we say we're at steady state with respect to time but not spatial position, at any given point in the crystallizer, if we look at the properties that we're interested in, we'll quickly go to a steady state in a tubular type configuration, particularly plug flow type configurations. And then if we are at a steady state, all of these average values should stay the same indefinitely. So this is what we would see in a plug flow as we go from initial primed solution to active crystallization at a given fixed point. So in order to see the actual progression of the crystallization with respect to spatial position or residence time, we can increase the length of the tubular section and take observations along this residence time or along the length of the tubular section if we keep the diameter of the tube constant. So this is fairly straightforward to do. You connect your routing type mixer to tubes of increasing lengths. You keep all the other conditions the same. And then you can make observations at increasing residence times, and you can see how your crystallization progresses as a function of residence time or as a function of spatial position along the crystallizer. So the previous graph showed that we do indeed have a steady state with respect to time. So at any one of these points, we'll have a graph that goes up from startup and then goes flat, and in theory should stay flat or with no movement in the average values forever, assuming we can sustain operation. And when we pull out those values at those flattened points, for instance in the last curve that we would observe doing each of these experiments, we can see that we have a decrease in the benzoic acid concentration, in this case done on a calibration-free basis. And also we can see that we've achieved steady state very quickly. So the residence time used here is on the order of 3 seconds. And in that 3 seconds, we're able to reduce down to our final thermodynamic maximum yield. And we would expect this to be borne out in the growth of the particulates. And we see, yes, that is true. We have nucleation and we do see growth as we go along in this 3 second residence time up to our final value, which has a mean chord length of approximately 20. In fact, actual particle size can be smaller than that. This is a representative measurement. It doesn't exactly translate to particle size when we use FPRM, but a very important qualitative tool in crystallization development. So we see this as a very simple empirical way to see have we maximized our yield. Is our crystallization length that we've chosen long enough for this high intensity plug flow crystallizer, although such an approach can be taken with any type of tubular crystallizer if desired. So we can compare the performance of the plug flow type crystallizer directly to the MSMPR crystallizer. So as in the previous slides, we'll look at how for this benzoic acid test system, the MSMPR starts off and reaches steady state. Again, I've shown this diagram already and discussed the pneumatic and intermittent way material is moved out of the crystallizer. So the pneumatic intermittent withdrawal of up to 10% of the vessel volume. So again, this was to prevent, first of all, fouling and blockage of this transfer line. So we saw in the phase diagram that the MSMPR always operates with some degree of supersaturation. So if we're moving material that's still crystallizing out slowly through this pipe, there's a higher chance of crystal growth on the walls, adhesion and cementing of particles to the walls, and fouling and blockage. But also we want to make sure that we remove at a relatively high velocity to try and achieve isokinetic withdrawal, which means that we aren't preferentially sucking out any of the particle size subclasses. And particularly if we're moving at too low a velocity, you can preferentially suck out the fines, leave the larger particles in here, which means that you're not operating like an MSMPR. So that was the driver behind this development of this pneumatic withdrawal mechanism. From the point of view of the dynamics, we can also very simply, as we showed already, put an FTIR and FBRM in situ and look at the concentration change and the change in the particle size characteristics via FBRM chord length measurements. And when we do this from startup, we have a couple of things to consider. So we can start up MSMPRs in a number of different ways. So one way that you might want to start off is you take your feed solution, you fill it up, maybe then you start to add anti-solvent, and you wait for the system to reach an equilibrium. So you can start off from clear solution. Similarly, you can start off with a batch, you put in feed solution, and then you add anti-solvent and you conduct a batch crystallization. Then you turn on your feed and anti-solvent pumps at the values that you want to set for residence time, and you then withdraw, and then you're in continuous operation. So these have different implications for startup. So typically you would be advised to wait up to 8 residence times for a MSMPR crystallizer to reach what is known as steady state, or no change in the mean values versus time. So if we look at the FBRM versus the number of residence times, and in this case 30 minutes was used. So 8 residence times is a reasonably significant amount of time to stand and wait. We see that we do go up from our initial batch conditions, where we have our square weighted mean or our particle size statistic and measure of the number of particles, the total counts that the FBRM is providing. We see that from the batch situation, we wash out a large number of particles and we have an increase in the average size within the crystallizer. And we see a little bit of an oscillation here. So what's happened is initially we're washing out our batch solution. We then begin to decrease the amount of surface area available for crystal growth. So we depress the surface areas, or the ability of the crystallization to remove the additional feeds and the supersaturation generated from the feed and the anti-solvent coming in. Hence we can undershoot the total size, but this is relatively minor in this case. In some cases, continuous crystallizations can have nucleation and growth kinetics in a way that they will consistently oscillate in terms of the number of particles and also the mean size over the course of crystallization, even if left to operate indefinitely. So we can enter these oscillatory steady states. In this case, the kinetics were such that we go to a flat profile here and here, and we see that it takes approximately 6 to 7 residence times. So literature would suggest about 8. And this is again to account for the fact that there's a feedback loop between the supersaturation or the concentration in the vessel and the population of crystals that exists in there. One way to bypass this long hold time would be to take From the end of a crystallization in a continuous campaign, the contents of the vessel, isolate them, and then throw them back in at the start of the crystallization and start with a solution that's very close to the steady state, thereby potentially being able to isolate from the first instance that you start your feed and your anti-solvent pumps, and not have this long time period where you wash out and you wait for the crystal population and the supersaturation to balance and reach a steady state. As I mentioned, that sometimes it's not possible to reach a steady state at all, and you will see oscillations in the mean size, and that may be periodic, going on indefinitely. One of the modes by which this happens is there's insufficient nucleation kinetics. So I should mention as well, the types of nucleation are very different in the agitated crystallizers, particularly the MSMPR type crystallizers, and the plug flow or batch. In plug flow or batch, at the start, we generate a lot of supersaturation, and then we get primary nucleation, followed by secondary nucleation, and then a growth-dominated region towards the end of the crystallization. Because the MSMPR is operating at a steady state with respect to spatial position and time, we really are at a true steady state. So at any point here, at any time, once I go to a steady state, I'll have the same concentration and the same particle characteristics at every point. But also it doesn't change with time. So it really just sits at that one point on the phase diagram that has a single mean size, a single concentration, a single anti-solvent composition. And so that is how we get the characteristics that we see in terms of the phase diagram. This means that we do have a large existing crystal population in here, and these are sufficient to both suppress primary nucleation, as the supersaturation can never get very high, so it can't be used to generate a significant number of primary nuclei. But also there is a population that can undergo secondary nucleation, which is typically contact mechanisms between crystals and crystals, or crystals and the impeller, or shearing of growing or assembling monomers or molecules on the crystalline surface that can be disrupted during growth and then can be used to form critical nuclei clusters at relatively low supersaturations. And so the secondary nucleation mechanism is what dominates here. The shear of the impeller is incredibly important for secondary nucleation purposes. The approach that I outlined briefly earlier, the use of a wet mill, essentially takes that out of the crystallizer and provides a lot more shear. So if you do find yourself in a case where you have oscillatory behavior that won't stabilize, the addition of a wet mill can drastically increase and alter the secondary nucleation kinetics of the system via both breakage of crystals and contact secondary nucleation mechanisms, and thus providing the ability to have a larger number of particles and higher surface area, and potentially kicking the system out of an oscillatory steady state into a stable steady state. That would be preferable. I think maybe a final thing to note with respect to the operation of an MSMPR in a single stage cascade or a single MSMPR. One implication of representative withdrawal is that we take very small nuclei that have just nucleated that might have a residence time of seconds, and we also have larger particles that have a longer time to grow that could have been in there for many residence times or many hours, just based on the probabilities of being removed in any given purge from the system. The implication of this is we have a broad particle size or crystal size distribution when we use single stage MSMPRs. And so one implication of breaking the anti-solvent charge into multiple stirred tanks is that if we have a third tank down here, so we go from one MSMPR instead of into the product collection into another and then into another MSMPR, we don't have the same spread of residence times for our products. So we have a narrower residence time distribution with the more stirred tanks that we add to the cascade. And so this is one consideration for using cascades in addition to single stage MSMPRs. So in addition to comparing the operation of continuous and batch, continuous MSMPR, plug flow, and batch systems, we'll look at their implication on product quality in terms of mean size, CSD, and morphology for this benzoic acid case study. So initially, we can look at the FBRM chord length distributions that were observed for the different types of crystallizations. And in this case, there will be an optimized bed batch for particle size optimization to get large, narrow particle size distribution. A reverse addition batch crystallization where feed is dropped into a large batch tank of anti-solvent to try and minimize particle size in batch operation. And this is compared to the high intensity plug flow crystallizer and the MSM-Pure crystallizer. So all the batch crystallizations had a batch time of 30 minutes in this case, and the MSM-Pure had a 30-minute residence time. So what we see here is first of all, from the particle size distributions, we see that we can get a wider range of crystal size distributions if we consider both continuous crystallizers and batch crystallizers. The two extremes are the MSM-Pure crystallizer and the plug flow crystallizer in terms of a very large number of small particles here. We see in the MSM-Pure we do have a significant amount of fines in comparison to the batch crystallization here, but also on average we do have a large particle size out here from the crystals that have had a long residence time in this relatively low supersaturation environment compared to the batch, which can build up significant supersaturation at the beginning. So I think this is important to consider. We'll talk about it a bit more. Another striking thing is the productivity. So we had about 2.9 kilos per hour productivity at this small plug flow crystallizer. These figures are for a 33 milliliter variant. So that was the retintype mixer with a short tubular section that had reached thermodynamic equilibrium. All of these crystallizations had approximately the same yield. The MSM-Pure obviously operates always with some supersaturation and hence some reduced yield theoretically compared to the batch and the plug flow. However, the benzoic acid system had very high nucleation and growth kinetics, and there was a relatively large anti-solvent charge, so we were quite close, or within sort of margin for error of measurement for yield in all these cases. So they are quite equivalent to compare. So if we go and we look at the comparative productivities of the crystallizers, we find that the continuous crystallizers are much more productive. So as just outlined, we have high productivity within this 33 milliliter plug flow crystallizer. So this will be equivalent in terms of output on an annual basis of about 80% capacity, so 300 days operation per year, to a 9-liter MSM-Pure crystallizer. So this becomes really striking though when you compare it to fed batch operation. So a 9-liter MSM-Pure crystallizer or the 33 milliliter plug flow crystallizer would have the same output on an annual basis as conducting 43.2 10,000 liter batch crystallizations at the same yield in the case of this crystallization. So we can see there's a huge increase in intensity, productivity, and this is seen across the board in many continuous operations. But you can imagine from a capital standpoint, a 10,000 liter fully installed batch reactor compared to a 33 milliliter continuous crystallizer that primarily consists of tubing and a route mixer, and even in this case the route mixer, since the study has been conducted, could be 3D printed in relevant materials such as stainless steel, titanium, or more recently PEEK. And so this really there is a paradigm shift in what's possible in terms of productivity and how we supply medicines. Microscope images of the product isolated from the runs shown on the previous slide are shown in this figure. So on the top left, we see the characteristic elongated plate shape with relatively high aspect ratio for benzoic acid from fed batch crystallization. One thing that is striking, if you compare this to the MSM-Pure crystallization, is that the plates have moved to this sort of step morphology and also 1-to-1 or close to 1-to-1 aspect ratio that really isn't possible to generate in fed batch crystallization in this system absent outside intervention, for example, with wet milling. The sizes are relatively consistent with what we've seen in the previous slide. And on the bottom left, we have the reverse addition crystallization, which was an attempt to drive down particle size to a minimum in batch, whereby the feed is added to a large amount of anti-solvent. While the nucleation kinetics are high for this benzoic acid system, the growth kinetics are also high. So in addition to small particles, some of the early nucleating particles at the beginning of the batch, at the beginning of the charge, are able to grow significantly in this environment and reach relatively large particle sizes, giving both small particles and some very large particles, so a wide distribution. It's very difficult to compare the plug flow output, so this is 10 times more magnification than the slides for the other types of product. To do this, it was actually better to look in situ, so using a flow cell to look at the particles in situ using the PVM flow cell. And in this case, just a very simple comparison is shown between the plug flow output and the standard anti-solvent crystallization, where you can see the extent to which particle size can be driven down via using these high energy, high supersaturation crystallizations. The emphasis of this webinar is on highlighting case studies that demonstrate that the critical quality attributes for API crystallization can be achieved within continuous crystallizers and outlining the popular or common crystallization strategies to do so. It should be noted that model-based design should be applied where possible for MSMPR type crystallizations and continuous crystallizations more generally. So pharmaceutical development timelines are more compressed than other industries, which creates a burden in terms of experimental load. MSMPR type crystallization experiments can be long, particularly if you have to wait several residence times for the crystallizers to reach a steady state. But in doing so, you can fairly easily derive kinetic information for that system and thereby put that kinetic information into a population balance model and to conduct a numerical optimization or computational optimization of all possible process parameters or all possible process conditions. And a couple of good examples of how to visualize and do this were presented by Vedder et al., whereby a system of using regions of attainable particle sizes, where particle size is the primary attribute of interest, primarily using kinetics and population balance modeling. This was extended by Power et al. in UCD and the SSPC to account for energy balance and volume constraints of the crystallization. And this was for a two-step cascade where essentially all of the possible mean square weights that could be achieved for the given crystallization system investigated could be predicted as a function of the total residence time that would be used, and there were additional constraints that can be placed. For instance, a yield constraint could narrow the range of particle sizes that can be achieved further. So this does two things. It allows operating conditions to get your product quality attributes that you desire to be predicted from a small number of experiments, but with a large number of computational experiments. But also it allows you to quickly evaluate based on a small number of single stage MSMPR experiments whether the product quality attributes that you desire can be actually reached with that platform, or if it needs to be augmented with the addition of continuous seeding or wet milling or something of that nature, or maybe a batch crystallization or plug flow crystallization would be more appropriate. So this is really key in pharmaceutical development, or the uptake of continuous crystallization in pharmaceutical development. And I direct you to these two papers as a starting point, or any of the textbooks listed in the reference section. Polymorphism is the ability for a compound, when crystallized, to exist with multiple crystalline phases or multiple crystal structures. So why is this important to control for the crystallization of APIs? Well, those different crystalline phases can have different solubilities, in your small intestine, which thus controls the bioavailability of the drug once dosed, for example, as a tablet in an oral solid dosage form. So as a result, we have to be able to control polymorphism in continuous crystallization if we are to use continuous crystallization techniques in the isolation and crystallization of APIs. So as outlined, there had been previous work prior to the recent spurt of development on continuous crystallization technologies for commodity chemicals. However, really none of these studies considered polymorphism, as these very small molecular weight, often inorganic compounds, did not experience significant polymorphism. So this was kind of an open question that had to be figured out through research, and ultimately the answer was yes, it can be controlled. And this is particularly interesting in the case of MSMPRs. So maybe to start, we'll look at a phase diagram again and look at the dynamics of the systems as we did previously for particle size control. So this is a monotropic polymorphic system, actually glutamic acid here, although not labeled. And these are temperature dependent phase diagrams. So as the temperature goes up, the thermodynamic or the maximum stable amount that can be dissolved in solution goes up. Hence the liquid phase concentration that can be dissolved goes up. So monotropic systems like glutamic acid, the phase diagrams or the solubility curves for the two polymorphic forms don't cross over as a function of temperature or as any of the other process variables that may be manipulated in this case. There is also an anisotropic systems whereby there is a flip over in the stability of the two polymorphs, usually as a function of temperature. So what happens in a normal batch crystallization, which also can exhibit polymorphic issues? So if we start off with an undersaturated solution, which will be clear, we then cool it down and we pass through the solubility curve for the stable form, in this case the beta form of glutamic acid. And we continue to cool. There's no nucleation to date. We're still in the labile region. We also pass through the solubility curve for the alpha form and the metastable form, or the unstable form of glutamic acid in this case. And eventually we either seed or we allow spontaneous primary nucleation to happen. And in this case, and in many cases, this nucleation favors the metastable form. So because there's higher primary nucleation kinetics for the alpha form, we'll see that appear first. Faster primary nucleation kinetics mean a lower induction time for nucleation. We then get secondary nucleation as these particles hit the impeller and each other and generate a population on which we grow alpha, more alpha crystals. So this way we can deplete the mass down to this first solubility curve. And in a batch vessel, we just leave this stir. Eventually, some beta form will nucleate as there is still supersaturation between the beta form and the alpha form solubility curves at this, which is the isolation temperature. And once this beta form nucleates, it will start to secondary nucleate due to the presence of supersaturation here. And this will then produce a population of beta form crystals, which will have some driving force. These will grow, sucking more concentration of the glutamic acid molecules from solution. This will bring the concentration below that for the alpha form, which will then dissolve, replenishing the L-glutamic molecules in solution. And this will occur until there's no alpha form molecules left, and then we will eventually move down to the solubility curve for the beta form, whereby the solution will be at thermodynamic equilibrium and isolation of a single phase of beta glutamic acid can be isolated. So continuous tubular crystallizers will follow the same dynamics that are seen in batch crystallization, again, except with the progression of the crystallization or the crystallization dynamics taking place over the length of a tubular section rather than over time in a batch vessel. So they're relatively easy to intuit how these would proceed. However, no one had really studied polymorphism in MSMPR type crystallizers. So there's a couple of things that we posited that would be can you isolate metastable polymorphs? So if we operate only at one point on the phase diagram, as we do with MSMPRs, the solution-mediated phase transformation that can make our metastable forms transform into our stable forms really doesn't exist. So if we stay with in a position that has supersaturation with respect to both the alpha form and the beta form, in this case for glutamic acid, there is no solution-mediated phase transformation possible in the same way that there would be in batch or a continuous tubular crystallization. So if we operate here, the idea is that we will operate in a state that is kinetically controlled. So essentially the secondary nucleation and the growth kinetics for the individual polymorphs will drive how fast they can take material from solution. And in such, there will be a competition between these two forms to see which will dominate within a MSMPR type crystallization. So this was the proposition that was made, and it did turn out to be an accurate way. And really, we want to see, can we isolate metastable forms and stable forms under this environment? And to look at this, we started as per the phase diagrams with L-glutamic acid crystallization. This is a common model system used in the study of polymorphism. And as outlined, 25 degrees, if you're going to do an isolation from a cooling crystallization, the metastable alpha form in this monotropic polymorphic system will dominate kinetically. So at 25 degrees, you see if you cool down to 25 degrees and hold, you only isolate API in the alpha form if you take a sample here. But slowly, over time, you do get some primary nucleation of some beta form crystals, which will nucleate, grow, take supersaturation or take concentration of L-glutamic acid from solution, thus dissolving the alpha form and eventually allowing a batch crystallization of only beta form crystals to be isolated. You can see this is very slow. It takes almost 40 hours for the transformation to take place. This can be temperature dependent, so the temperature dependent kinetics of each of the forms may vary. So if you isolate or hold at 45 degrees, you'll see that initially in the nucleation event, which occurs here, there is some beta form from the beginning, and hence the transformation is much faster, on the order of 7 hours compared to 40 to go to complete transformation from a mixed population in this case to a population of crystals that is entirely beta form crystals via solution mediated phase transformation. So we see that the secondary nucleation and growth kinetics for this process is more favorable for the stable monotropic form at higher temperatures, 45 degrees in this case. And we see in the batch crystallization, we only get nucleation of the alpha form and a very slow transformation to the beta form or the stable form, which would typically be sought in API crystallization processes at 25 degrees, which incidentally, as it's a lower isolation temperature, will yield a higher yield as the solubility is lower at this point. So this was investigated in this paper, which outlines for the first time how to control polymorphism in these types of continuous crystallization systems. And we ran the MSMPR experiments at lower temperatures. Only alpha form was isolated in the crystallization runs for MSMPR systems. And this is at a variety of residence times up to 120 minutes. But when the MSMPR crystallization was run at 45 degrees, it was possible to directly isolate the stable beta polymorph of L-glutamic acid. So this was the first demonstration that was possible to isolate both alpha and beta polymorphs, so metastable and stable polymorphs in MSMPR systems. However, this begged a couple of questions. So first of all, were these steady states stable steady states? So we talked already about how you get oscillatory steady states that would go up and down. So are these steady states that are achieved in the crystallizer going to continually and perpetually produce pure beta or pure alpha, which would be desired for use in API crystallization? So to test this, we conducted an experiment whereby batch crystallization was conducted and this was allowed to progress to transform to produce a population of entirely beta crystals, so the stable form for L-glutamic acid. And the question was, if we have a seed loading that is very, very high to start off in MSMPR crystallization, that is the stable form, would the stable form persist, or would we go to a stable or a kinetically stable steady state that produces the metastable form? So essentially, is this a self-correcting and stable steady state that allows us to produce a metastable form at scale, which would be a development and potentially useful in a variety of industries? And the answer was yes. So after the batch crystallization, the pumps were started up and crystallization proceeded, and a steady state that was metastable for the stable form of the API was achieved. But once some alpha form was nucleated at this point, the alpha form began to kinetically dominate. So at this temperature, it had more favorable secondary nucleation and growth kinetics. So it was gradually able to go back to the same steady state that was achieved in the previous runs. So this confirmed that yes, it is possible to isolate metastable polymorphs continuously, and also that these are kinetically stable steady state, so they will self-correct even if we put in a huge seed loading above a 100% seed loading relative to the steady state for the crystallization of the stable form. We will revert back to the metastable crystalline form and isolate only alpha again if we run this crystallization at 120 degrees, 120 minutes at 25 degrees. More broadly, in continuous crystallization systems, if you're concerned or you want to verify that your steady state is a kinetically stable steady state that will be suitable for manufacturing, and you want to do this empirically, competitive seeding experiments would be a way to do this. So going to a steady state and seeding with an alternate polymorph to see if that will go on to dominate, or as in this case, at the most extreme case you can do, have a very high above 100% seed loading and allow the crystallization to the opportunity or enough time to self-correct if it has the tendency to do so back to the original steady state at which you've selected for manufacturing. So this study that I was involved in Alan Morrison and Bernard Trout's research group in MIT developed a lot of the tools for developing continuous polymorphic stirred tank crystallizations. To date, in the slides demonstrated that it's possible for the first time in this study to isolate metastable forms in this way at scale, and also the thermodynamically stable form, which in this case was carried out by operation at 45 degrees. However, if we look at per pass yield in a continuous crystallization system, obviously operating at 45 degrees to isolate the stable polymorph will result in a lower yield. With kinetic modeling, population balance modeling, it can be evaluated very quickly. What would the residence time requirement in a single stage crystallization be in order to isolate down to detectable limits only the beta stable form at a 25 degree crystallization and isolation temperature? If you can see, this is over a 900-minute residence time, which is very long and is approaching something very similar to a long batch crystallization. So this would be fairly pointless and not a strategy where you would usually pursue crystallization, particularly for manufacturing purposes. What would typically be done would be higher temperature crystallization, but use a recycle loop to make sure that no yield losses are incurred for operating at these higher temperatures in order to control the form of the API that is derived from the crystallization. Because MSMPR systems control the polymorphic content within the crystallizer at steady state through kinetic competition between the two forms or three forms present or are possible for a polymorphic system, there is no extra considerations really for enantiotropic systems compared to monotropic systems, as it's really the kinetics of the system that controls the output rather than the thermodynamics, as would be the case in batch crystallization. So this was found to be the case, and a follow-up study here shows the phase diagram for paraaminobenzoic acid. So this enantiotropic system has a high temperature stable form, which is an alpha form, and it has a low temperature stable form, which is called the beta form. So previous batch crystallization study demonstrated how difficult it is to produce the beta form of paraaminobenzoic acid through batch crystallization. So the primary nucleation kinetics are very slow for the beta form of paraaminobenzoic acid, and very difficult to isolate as a batch from a batch crystallization. As a result, the commercial form that you get when you order is the alpha form. So one surprising thing about running this system continuously in an MSMPR for the first time is that the beta form dominated and was by far the easier form to isolate in contrast to all batch studies conducted. So this is due to the fact that the beta form, while having very poor primary nucleation kinetics, so to nucleate in batch was difficult, had good secondary nucleation and growth kinetics and was able to kinetically dominate the alpha form unexpectedly. But the study here posed the question, what if we do want to continue to isolate this alpha form, that's the commercially available form, using continuous crystallization? And also to attach an additional constraint. So 90+% yield, which is fairly typical for very well designed cooling crystallization, and also no detectable beta form. So this is 95% polymorphic purity in this case, which was the detection limit for X-ray for this polymorphic system. And again, the residence time is capped for a two crystallizer cascade system at 120 minutes. So it was possible to vary the residence time in both and the temperature of both at which they operated at their steady states. And this was done numerically. So this was again done by population balance modeling. And in the results of the study showed that there was regions of attainable polymorphism and yield. And there was a segment of the overall landscape in terms of the crystallization dynamics and possible outcomes that did allow above 90% yield and no detectable beta form to be isolated out of the product from the second crystallizer. Again, emphasizing the importance of kinetic modeling in development of continuous crystallization of APIs, as it would have been very, very laborious to do this study purely empirically to find out if there was a possible operating region that can meet the polymorphic specifications for a given system. So following these studies that showed how to design continuous crystallizations to isolate either stable or metastable polymorphs in a way suitable for manufacturing in MSMPR type crystallizers or their cascades, Mike Doherty's group in UC Santa Barbara did the higher level systems engineering work, and for two polymorphic kinetically competing systems in MSMPRs, they were able to reduce down to 3 parameters based on dam color analysis, a prediction of which polymorph, if the kinetics are available for the crystallization of either of them, will occur at steady state in a continuous MSMPR type crystallization system. So taken together, this will essentially allow prediction of the output of MSMPR type continuous crystallizers, which again now provide a way to manufacture both stable and metastable polymorphs in a scalable way. I didn't discuss co-crystallization or the crystallization of salts here. These mechanisms will apply where there's polymorphism in systems with salts and also co-crystals. There will just be an additional design parameter for weaker salts and also co-crystals where the counter-ion or the co-former that's added to the solution, the stoichiometric excess of that will need to be controlled as well. So there's actually an extra design or control parameter to manipulation of co-crystallization or salt systems where there are polymorphs, there will be a similar or the same controlling kinetic mechanism at play for MSMPR systems for their crystallization and continuous isolation. So the final thing that we'll discuss in terms of product quality attributes needed for APIs and how continuous crystallization can deliver these will be purification and continuous crystallization, or to demonstrate that continuous crystallization can meet the purity requirements that are so rigorous and required for drugs to be released and used for human consumption. So currently in batch pharmaceutical production, crystallization is the most commonly used purification technique. So it first of all allows for high levels of discrimination between APIs and impurities. It is highly volumetrically efficient. It is relatively inexpensive to conduct. And also, obviously, it simultaneously allows for final API product quality attributes to be controlled during final API isolation. It also allows for intermediate isolation and stockpiling of material, which has a number of benefits in terms of the batch supply chain for pharmaceuticals currently. There is, however, relatively little material in the literature on continuous pharmaceutical crystallization for the purposes of purification, although there are a couple of notable studies. So the batch crystallization methodology is used. A slow batch crystallization conducted over many hours unagitated is quite a powerful purification technique, as mentioned already. However, when you push crystallizations to go faster and faster, so if you conduct them at lower batch times by accessing higher supersaturations, typically or commonly, the purification efficiency of that continuous operation can be driven downwards. So the ability to purge impurities is reduced in the case of some crystallization systems if higher supersaturations and lower batch or residence times are used in the case of continuous. As we've seen in continuous operation, we will use, or at least try to use, relatively low residence times in comparison to batch crystallizations, which have to be slow due to their need for a large amount of scale up in batch operation due to lack of productivity in that mode of production. So one question is there sufficient ability to purge problematic impurities that have to be controlled to almost uniquely low levels in API crystallizations? So to this end, I'll highlight just a couple of studies. Again, another nice study from Professor Alan Morrison and Professor Bernard Trout's research group as part of the Novartis MIT Center for Continuous Crystallization for Continuous Manufacturing. In this case, cyclosporin, a relatively large molecular weight small molecule API with a molecular weight of 1,200 was utilized. The system has a lot of impurities that are structurally very similar. Such impurities are often referred to as lattice substituting impurities, as they can partition into the crystal lattice of the product as the crystallization occurs due to their structural similarities, which creates a relatively low energy barrier or means of or discriminatory ability by the crystallization process. So being able to purge structurally similar lattice substituting impurities is a high bar that continuous crystallization has to clear in order to be able to be utilized for continuous pharma manufacturing. And in the next couple of slides, I'll present a couple of strategies whereby this can be done. In the case of cyclosporin, the batch crystallization, if allowed to proceed for the same amount of time as the equivalent single stage continuous crystallization, so 3-hour residence time in this case, can get to a 74% yield with 95% impurity. So this will be considered a relatively difficult system to purify in batch crystallization. In the case of the MSMPR, so using MSMPR cascades, it was found that the purification efficiency was reasonable, but the yield that could be achieved at this level of purity was relatively low, and this required a 3-hour residence time continuous crystallizers, which obviously has a lot of residence time, long startup times, and has a lot of additional things to consider compared to a batch crystallization. The main reason why the yield was so low for both of these is that at low temperatures, this relatively large molecular weight, small molecule API, it's very slow growth kinetics. So to get up to the approximately 90% yield that you would try and target for cooling crystallization, this is very difficult. But in batch, you need a long hold time in batch, or you need a lot of vessels with relatively long residence times in multi-stage cascades or MSMPRs. So given that we tend to want to run relatively short residence times for continuous crystallization operations to access the higher productivity that we would seek in a move to continuous manufacturing, but also to be compatible with high productivity flow chemistry steps upstream of a continuous crystallization process, for example. So to deal with this challenging case, or really any case, it's possible to utilize recycle loops. So in this case, you can take a continuous crystallizer that is fed with a feed solution. You withdraw your mother liquor and your solid phase out. You can take out your solid phase using a filter, which is purified at this point, and the mother liquor can be collected. So rather than discarding this mother liquor as in a standard batch crystallization, this can be, in the case of cooling crystallization, can be evaporated, or at least partially evaporated, to bring it back to the feed concentration, or to partially reconcentrate it and fed back in as a recycle stream back into the crystallizer volume. So in this system, really, we can get to 100% yield. So if we don't purge out any of our mother liquor, we have a 0 flow rate to waste in the system with recycle. The only way for material to leave the system is add solid phase product as gas. So some of the solvent can leave as gas. So really, in this case, there's no waste, or everything that can crystallize does crystallize. This inherently has a downside in that if we don't have a purge stream here, any impurities that are non-volatile will crash out and be removed with the solid phase product. And this would very much include the lattice substituting impurities that would partition both into the crystalline product for cyclosporin, but eventually, if they reach high enough concentrations, they'll precipitate out and form their own solid phase that would be mostly impurity. So really, in continuous crystallization, we can run our crystallizer at a steady state that mightn't have the highest yield, but we can utilize recycle strategies in order to maximize yield and purity trade-offs. While this does give you better yield and purity trade-off, it does reduce productivity if you use very high recycle rates back into the vessel. So this is something to consider. You have a trade-off between productivity, yield, and purity. And in the case of cyclosporin, for example, this allowed, instead of a 3-stage MSMPR cascade, to move to a single-stage cascade, but go up to 91.8% yield while maintaining equivalent purity for this very challenging API impurity system. So just demonstrating that high yields and purities can be achieved in continuous crystallization. So the recycling strategy outlined would be very effective for cooling crystallizations, where concentration by evaporation won't radically affect the composition of the mother liquor. However, we'd like to be able to extend this to anti-solvent and other types of crystallizations, and to this end, organic solvent nanofiltration can be employed instead of evaporation for concentration prior to recycling of mother liquor. So very briefly, organic solvent nanofilters are solvent-compatible semi-permeable filters that are able to retain molecules that have a molecular weight on the order of small molecule pharmaceutical intermediates, ingredients, and products. So in order to demonstrate that the integration of nanofiltration can allow mother liquor recycling techniques to improve yield and purity in continuous API crystallization that rely on anti-solvent or mixed solvent systems to be conducted, an API example crystallization was conducted that was a combined cooling anti-solvent crystallization from a mixed solvent system in order to isolate the API deferrocyrox and purge an impurity for hydrazinobenzoic acid, which results from its synthesis down to very low levels. So this is both a lattice substituting impurity, and it also must be cleared to a maximum below 4 ppm concentration in the final solid phase deferrocyrox product. In many cases, though, this is controlled to a much more rigorous level in the drug itself. So to do this, we take the previous system where we would have an evaporator and a recycle loop set up with solids removal, mother liquor collection and concentration, and add in a membrane module in order to conduct the concentration. So some of the material will permeate the membrane, and this will go to waste. One interesting thing to note, if the membrane is permeable for the impurity and not permeable at all to the API, the system will not need an initial purge stream and will operate effectively with 100% yield and 100% purity. However, there's always at low levels other impurities in there, so there will be a necessity to do some at least limited purging. But the question in conducting this study was, would it be possible to go to much higher levels of purity using membrane recycling techniques? So in the case of this deferrocyrox demonstrating system, the answer was yes. So the results are shown here for batch crystallization, which could achieve a yield of 90% and reach down to 0.32 ppm level concentration in the product of 4-hydrazinobenzoic acid. A single stage MSMPR that had a residence time set to be the same as the batch time for this combined cooling anti-solvent crystallization would have a yield of only 70%, but at this reduced yield there was a low level of impurity or a lower level of impurities found. Again, this is right at the limit of detection for the HPLC method that was available for release. So these are effectively equivalent but very high levels of purification. When a recycling strategy with two different membranes was included, it was found that the yield purity trade-off could be greatly improved. So the yield could be pushed up to over 98 and over 99% while maintaining purity in excess of what could be achieved in the batch crystallization. So the graphs will show the attainment of the concentration in all of these streams versus time as the system as a whole goes to steady state. And just to note, the purge stream here in the crystallization without membrane has equivalent to concentration at steady state to the other streams as it should. But if we look at the permeate from the membrane, has a much reduced level of API getting out of the system. So modeling studies showed this could actually be pushed over 99%, but this was good enough for any industrial process. If it could be achieved, you would be very happy with this. There was further studies that used addition of complexing agents that did couple with the impurities to allow their preferential removal, even when the membrane wouldn't be able to discriminate between APIs and impurities. So a generalized technique for complexation and removal of problematic impurities was formulated in follow-up studies in the literature to this one. So finally, just to sum up, we have gone through different types of continuous crystallizers. We've spoken primarily about the most common platform used in the pharmaceutical industry for continuous crystallization, MSMPR type crystallizers and their cascades. talked about how these different continuous crystallizers have different dynamics and how these in turn can be used to control all the critical quality attributes needed in API crystallization. Maybe a small omission, we didn't discuss the subcategory of purification of chiral resolution or removing enantiomers, as most synthesis is conducted asymmetrically in pharmaceutical synthesis. In many cases, this is more common this is more comparable to the removal of a standard impurity rather than a full chiral resolution from racemic conditions. However, I've attached a couple of references for both of these, but won't have time to discuss them today. I'd like to briefly as well thank the various research groups I've been involved in, my own current research group, in addition to Brian Glennon's research group, where I did my PhD and who I've collaborated with since returning to UCD as an academic, Professor Morrison on Trout's research group in the Novartis MIT Center for Continuous Crystallization, where some of this work was done, and also to all other collaborators I've worked with throughout the years. So thank you very much for your attention. I'll briefly pop up the extended reference list and take any questions that you may have at this point. So thanks very much.